Welcome to Science Philosophy in a Flash, a mini podcast series produced by the Scientist Creative Services team. In this series, we highlight researchers' unique outlooks on what it means to be a scientist. Xitiz Chamling is an assistant professor of ophthalmology at Johns Hopkins University School of Medicine. From vision research to the pursuit of multiple sclerosis treatments, his philosophy of science centers human-based systems to study the neuroprotective layer called myelin, which surrounds and insulates nerve cells. In his latest work, Chamling combined advances in stem cell and CRISPR-Cas9 technologies to break away from conventional high-throughput drug screening platforms that rely on rodent cells.
During my PhD, I got into vision research, looking at vision defects and blindness. So that led me to Dr. Donzak's lab at Johns Hopkins, where he was using human pluripotent stem cell as a model to study vision defects and vision research. Vision and all the information is formed in the eye, but it has to be transmitted to the brain. There is a bundle of retinal ganglion axons that go along the back of the eye to the brain. It needs to be myelinated. If the myelin is damaged, the information is not transmitted and the vision is lost. There is a lot of other neurological diseases that are because of the loss of myelin. So this whole idea of studying myelination to find a way to repair myelination in the optic nerve kind of led to a bigger picture of looking at multiple sclerosis. What happens in multiple sclerosis is your immune system thinks your myelin is a foreign material and it attacks and degrades it. A lot of people like to call it is like a mistaken identity. If you suppress the immune system from attacking your myelin, you can prevent the progression of the disease. But just by stopping the immune system, you can't recover the myelin that's degraded. There is a huge interest in this field to reestablish myelin, which is called remyelination. One of our thoughts was, if we are doing drug screening, what if we start by using the human cell? Probably wouldn't have been possible if it was, even like 10 years ago. Prior to that, even making the human cells to use for drug screening was hard to imagine. First of all, we need a lot of cells. Another reason why it would not have been possible even 10 years ago was whenever we were doing a drug screening, one key aspect is editing the genome like we do in a genetic mouse model to label the cells and use them as a readout to validate or to confirm that the screening worked. And finding a way to do that in human cells also was very difficult. Being able to combine this genome editing tool with the stem cell technology really helped us build a platform where we can use human cells and to read the effect of a compound in a much more simpler way.
Thank you for listening to Science Philosophy in a Flash. To learn more about Chamling's research, please check out the link in the episode notes.